Hello, ladies and gentlemen. Welcome to Chapter 13A. In this chapter, we are specifically going to talk about protists. Basically, all those little things that are floating around in the pond water. Hopefully, not in your drinking water. Okay? Okay, the groups of protists that we're going to talk about. The first group is called protozoa. These are the animal-like protists. In other words, they do things kind of sort of like animals. Mostly they do. And the second group are the chromista, which are plant-like protists mostly. Um, what the mostly means for both of these groups is that there are creatures that don't really fit into either group, so they have been placed into a group and they kind of fit there, but they have exceptions and they don't really fit there, but nobody wants to make a whole new category in order to talk about them. So you'll see some exceptions both for protozoa and chromista. Okay, now characteristics of these protozoa, the animal-like protists. First off, unlike the prokaryotes, they are eukaryotes, okay? So they're not like bacteria, they're not like the archaea. These do have membranes, they have the organelles with membranes, so they have the mitochondria, the endoplasmic reticulum, nucleus, all of that. Okay, so all the cell structures that you learned about, they are there. Okay, secondly, they are usually, notice usually, motile. That means they can move themselves. Okay, kind of like bacteria with the flagella, they can move around. They are not usually going to be just floating from place to place, although some things are more likely to. Okay, third, just like the prokaryotes, they are unicellular. They're made out of one cell. But what's different is they also can live in colonies. And we will talk about what that means when we get to some examples of what a colony is, although you probably have an idea already. Okay, they are mostly heterotrophs. Hetero, remember, means other. Troph means to feed. So these are things that are going to eat something that is not themselves in order to get energy. In other words, they're not going to be making their own food like a plant does. Mostly. Okay. Next, they will need to live in water. This can include the oceans with salt water, fresh water, and even in small amounts of water that are found in the soil. So they don't have to be swimming around like you think of for a fish. They just need to have that dampness and that moisture. Okay, primarily, protozoa are characterized by how they move. They do not have nice simple names, but you're just going to have to learn what goes with what. The first category are called the sarcodines, and they use what are called pseudopods in order to move. We're going to explain all of this when we get there. So just realize sarcodines, pseudopods. The P is silent. Okay? So it is pseudopods. Second group, mystigiophores. Gotta love that name. They use flagella to move. Flagella you should already know. Third group, ciliophores. They use cilia. It actually makes sense that they are ciliophores. And the last group is a relatively new group, which are called the Ampicomplexa. Ampicomplexa. Okay? They realized these things aren't really in any of the other groups, so they did make a new group. When I went to school, these things, at least this category, did not exist. And you'll see why they're different than the other three groups when we get to them. They primarily will glide to move. And in reality, they don't know that much about how they do that yet. So you're not going to have to learn that much yet. Okay, now the sarcodines, an example of these would be the amoebas. Okay, if you've never heard of an amoeba, this is kind of what one looks like. You can see it in the still shot on the video. And these are the pseudopods that we're going to talk about. Okay, you can see these kind of things sticking out, and you'll see that they're going to change shape and size and all of that. And what's happening, if you look at this upper right picture, is the inside is going to flow in one direction. It's going to kind of create this current going one way. And when it pushes out, that thing is called a pseudopod. And it actually means fake foot. So here you go for the video so you can watch how it moves around. Okay, you can see through it. You can see the cytoplasm or plasmasol moving through. You can see on the edges of it, it's a little bit clearer. It's kind of the edges of the material, and you can see that it's actually moving across as it goes. It's pulling up one side and stretching out the other side. And it's doing this by moving the inside. You can see that current. And realize this is three-dimensional. That's why some of it looks blurry, as it's going into different levels. The microscope can only focus on one level at a time. Okay, now, this is kind of a still shot, and what you can see is this is the same process. The process is phagocytosis, in other words, eating. And what you can see is this pseudopod right here, sticking out. It's actually reaching around the food. And you have these lysosomes, if anybody remembers what they are. Lyso, remember, means to cut or break. Some just means little object or little body. These are what are like little bags with digestive enzymes inside. They're going to break down food. So those pseudopods you can see in the next picture, they have surrounded the food. It ends up becoming a vacuole or a bubble. These lysosomes are going to combine with that vacuole, the bubble, and they are going to digest the food. Okay, that process is called phagocytosis. You can see it over here. And it just means eating. The phago or phage, depending on the word, is the PHAG means eat. The cyto means cell. Okay, so this is eating by the cell. Okay, and you end up with all the food for the cell being digested. And like I said, this is kind of like an animal, so for the amoeba, this would be kind of the equivalent of its stomach going through. Okay, now this video is going to show you phagocytosis. It is sped up 2 times. It's not a fast process, but when you eat, it isn't always that fast either. Okay, you can see the little microorganism. It's kind of stuck in there. It's not really bright. But the pseudopods reach around and they completely surround that poor little paramecium, who is now officially in a food vacuole. Okay, you can see it is trapped and it will end up becoming food. The lysosomes will come to that food vacuole. You can see it's closing in. And in time, that little paramecium becomes amoeba food.